Excellent. So let me start introducing the speakers. But first, I want to tell you what is the goal of this panel. And this panel is different both in content and in composition from what you've seen so far. So in the first part of the conference, it was primarily dominated by AI researchers who developed new models and told you how they believe these models can change clinical care. And we also had from some clinician, in particular Dr. Lehman and from Dr. Klintz about how they're using these techniques in clinical care. So the goal of this panel is actually to talk to people who are really facing the pandemics and who are handling the crisis, and to hear from them what are the questions that we are not addressing. So it's kind of to see what is their experience and what we can do in the future. And I know that a lot of participants come from AI community like myself, and we always ask ourselves the question, do we solve the right problem? So hopefully after this panel, we will have a lot of new ideas of what, what questions we should be addressing and what are the real limitations. And I would also ask the participants, please feel free to ask questions, because I hope that it's going to be really interactive and we would like to hear from you. But with that, I want to start actually letting our participants introduce themselves. So I would start with Dr. Raj Gandhi from MGH.
Sure, it's a pleasure to be here. Thank you for inviting me. I'm an infectious disease physician at Mass General. I'm a professor of medicine at Harvard Medical School. And my focus in the past has been on HIV, but in the last 6 months, mainly focusing on developing treatment guidelines for COVID-19 here at Mass General, but also with the Infectious Diseases Society of America, the National Institutes of Health, and conducting treatment trials. And I have been involved in treating people with COVID-19.
Thank you. Thank you. So let's go next to another medical doctor, Guillermo Torre from Tech Salud, if I pronounce it correctly.
Yes, thank you, Regina. I have the responsibility of overseeing the healthcare division of a large private university in Mexico. We have 2 private hospitals. I am a cardiologist and an immunologist, and I spent 25 years doing academic work at Houston Methodist Hospital and had an appointment at Baylor College of Medicine for many years. But my work today in Monterey is to oversee and give a strategic development and confronting our system with COVID. We were facing a very unique situation in Mexico where we are in a private sector that provides care for a small percent of the population. And our strategies and policies were drastically different to the state policies. For instance, very little testing at the state level. We took a very approach for testing. We had ample resources in beds, ICUs, ventilators, and the government didn't. So we had to reinvent ourselves and to provide service not only to our community, to the country and the city at large. And so I've been taking care of patients and setting policy for a private institution that took the responsibility of extending care to the larger community. Thank you for inviting me, Regina.
Thank you. So our next speaker is Dr. Karen Wong from CDC.
It's a pleasure to be here. My name is Karen Wong. I'm an internal medicine physician and the data science lead of CDC's machine intelligence and data science team. And I practice as a hospitalist at Emory University Hospital and have treated COVID patients there. As far as COVID, starting in January, I was deployed to CDC's pandemic response, and I've been the data science lead for CDC's COVID-19 clinical team.
Thank you. Thank you, Karen. And now we will have Gabriele Antici speak, and she is another non-medical doctor on the panel, so we are minority, Gabri.
Thanks, Regina, and thanks for the invitation to participate in this great panel. So I'm Gabi Antici. I'm the founder of the Protea Institute. Protea is the first NGO that gives access to breast cancer treatment to underprivileged women in Brazil. I live in Brazil, I'm Brazilian. I'm also 1992 graduate from the MIT Sloan School of Management. I have had a career in finance. However, my life really changed forever. 4 years ago, when I was first diagnosed with breast cancer, I am a breast cancer survivor. I had breast cancer twice in the last 4 years. And because I had access to not only screening, but also treatment, immediately I had the chance of cure. And unfortunately, in Brazil, the majority of the Brazilian women, because they don't have health insurance, they don't have the chance of cure that I had. And for this reason, I founded the Protea Institute. And for this reason, I met Regina, I met Connie, and we have very promising work in Brazil in the future.
Thank you. And even though some of the audience may be wondering how it relates to COVID, but there is clearly a very strong connection, because as Connie was telling us just a few minutes ago, that, you know, the chronic diseases like cancer didn't go away. The screening facility, the treatment facility now operate under the reduced capacity. And we hope to hear from Gabriele, which will tell us more how it impacted, you know, the treatment of the underprivileged women in the new pandemic situation. But let me start with a few questions. So I would tell you that when we met before the conference, I didn't have a chance to read these poisonous, confusing articles that say how AI failed the pandemics. I strongly feel it's wrong, and I hope that after our panel it will be clear to everybody. But let's just start one by one. So I don't know if you had a chance to see multiple presentations, but you can see there were presentations of how to monitor the patients, how to, you know, do the modeling of the spread of the disease, predict possible, you know, issues with like complications. My question is, I want to start with Raj and ask him, what are the crucial areas in COVID care, management care, that were not addressed, that you think that really data science has a huge opportunity here and we are not working on it?
Great, so thank you for asking that. I'll tell you where my focus has been, and I do think it's an area that's ripe for AI. So my focus has been on treatment and developing guidelines for therapy. And when I think about COVID, I think of it in multiple dimensions. And I know people in AI like to think in multiple dimensions. I think about the features of the infected individual. We heard a fair amount about respiratory illness, but we all know that severe COVID-19 also is a multi-organ disease, from neurologic complications to blood clotting to cardiac or heart manifestations. And there's this idea that has become quite current of thromboinflammation, this idea of thrombosis or clotting plus inflammation. The second dimension, and you'll see why this is important, is what is the stage of disease? How severe is the disease? Some people, as we heard earlier, never become symptomatic, but of those people who become symptomatic, put some numbers on it, about 80% will have mild to moderate disease, 15% will have severe disease, end up in the hospital, and about 5% will have critical disease. And then lastly, and this is where I'm most focused, is the intervention itself. Our conceptual model for COVID-19 is that antivirals are likely to work early in the course of the disease. Anti-inflammatories are likely to work later in the course of the disease when people have this hyper-inflammatory response. So I do think that there's this multidimensional challenge, and I think we don't precisely know how to best target therapy. And let me give you a couple of examples, and then I'll pose some questions for the field or for your participants. So right now in a hospitalized patient, we give the antiviral drug remdesivir. This is known to shorten the time to recovery, and we give the anti-inflammatory drug, which is kind of a sledgehammer, called dexamethasone, which has been shown to improve survival. But we only know in very broad strokes how to use these drugs, very general criteria. Essentially, if someone needs oxygen, they're in the hospital, but that's an extraordinarily heterogeneous group. What has not been done, and what I think needs to be done, is to integrate more sophisticated physiologic, laboratory, and radiographic data. Very little has been done with viral levels, and then inflammatory markers. We also don't know, and for the first time, we're beginning to get some insights into combining antivirals and anti-inflammatories, but we really don't know how to do that. The field is just too new. And the urgency is only going to increase because now we have more targeted antivirals, monoclonal antibodies, for example, convalescent plasma. We have more targeted anti-inflammatories. Dexamethasone is a sledgehammer. It shuts down the immune response very broadly, but we have JAK inhibitors, cytokine blockers like interleukin-6 blockers. So what I would pose for the AI field, for all the people on this call and as well as the community, is can we get more refined and sophisticated approaches to deciding when to use different drugs and how to use them in combination? Can we integrate physiologic data? The Emerald project from earlier was an interesting idea. Laboratory and radiographic data, Dr. Eldar's data, when deciding how to use our drugs. And then can we not lose sight of the fact that most people with COVID-19 are not hospitalized, and what is the best approach to those individuals? So I do think randomized trials will give some of the answers, but I wonder if AI can be used to inform the design, analysis, and interpretation of those trials. And if you'd like, I'll give you 3 examples of patients that I'm seeing right now that I think could very much benefit from this kind of approach. So I have a person who's 90 years old, who I called his son last night. He's sitting at home. He's not on the Emerald system, unfortunately, but his oxygen saturation is a little bit lower than normal. It's about 93%, 92%. Should he be monitored at home? Should he come into the hospital? Should he get a monoclonal antibody? There's some promising data from a monoclonal antibody from just a week or so ago. A second example of what we don't know what to do is a 30-year-old man who was admitted a few months ago, severely ill with COVID-19. He developed mesenteric ischemia, essentially thrombosis and clotting of his blood vessels to his intestine, and essentially had ischemia and death of his intestine. Why was his course so severe? We don't know. What was the intervention to use? Should it have been anticoagulation? Should it have been an anti-inflammatory and antiviral or combination therapy? And then the last concrete example of a problem that we don't know the answer to is a woman who's had a kidney transplant, is immunosuppressed, and presents with heart failure, has evidence of edema or swelling in her myocardium. In some other instances, anti-IL-6 therapy has been given, but what is really the approach? And so these are just 3 examples of how our treatments right now are very broad, heterogeneous populations. We really don't know how to personalize or how to give optimal therapy. And this multidimensional challenge, I think, is a critical one to solve. So I'll stop there with questions for the field. So.
Thank you. Let me just ask a few questions.
Sure. Yeah, of course.
Sorry for monopolizing. I guess that's what happens when you run the panel. You can. So my first question, it's actually what you said is really interesting. How do we target therapy based on like rich phenotypical description of the patient? Yeah. And really for the AI community to answer this very pertinent question, one needs to have the data. Yeah. And this data doesn't have to be with patient names. It can be totally anonymous, of course. We can just represent every patient with some list of, symptoms and preconditions and medications. How far do you think is the medical community, let's say our Boston community, from releasing this type of data to, to the AI field so that one can make the models that you are eager to use? Yeah, so we did collect, without knowledge as to whether it would be useful, fairly systematically, a relatively large number of data elements. We obviously know things like vital signs, oxygen saturation, ventilatory support, but we also collected things like interleukin-6 levels, which is a marker of a cytokine that's, we think, is correlated with poor outcome, C-reactive protein, an inflammatory marker, D-dimer, a thrombosis marker. So we have these data sets, and they're relatively well curated alongside clinical data. How did the person do? When were they discharged? Did they survive? Were they left with any complications? Although the longer term data, how are they 6 months from now, is still in its infancy and not really well collated. But we do have those data sets, and I think at least the Boston hospitals were relatively uniform in collecting some of this information. In a pinch, we really designed a standard treatment approach, and part of that included collection of data. And so I do think after this call, we could connect and point out what are the data elements that have been collected. And we have a clinical outcomes team that I can point you towards, who have assembled these data into, I think, a usable format. So this is fantastic, because I can speak for the whole MIT team and for all our students that are so eager to contribute. So after this your panel, I will contact you, and hopefully we will find a way to to give this data to those who can make predictions that you guys can bring back to the clinic?
I think the big trials, the most successful trials have been large trials, which were not done with a lot of physiologic biomarker data. I'll give you the example in the United Kingdom. They did a trial called Recovery, which led to the adoption of dexamethasone. They did not collect physiologic data beyond supplemental oxygen need. And even there, they didn't distinguish between low amount of oxygen and high amount of oxygen. They just lumped it. They didn't collect the data on biomarkers. But some of the more targeted trials have collected some of that, viral levels, for example, inflammatory markers. And I think another rich source of data would be clinical trials that have done that kind of work. And we, again, have connections with that. So
I think this would be really, really fantastic if we can take all this data and get some really new insights that you cannot get by using like statistical methods that are typically.
Right. They have biostatisticians, but they don't have this level of, you know, they have very good biostatisticians, but not the type of work that your field does. So.
Great, you know, one can combine different strengths, and at the end what counts is the quality of the prediction. Now, if I may ask you another brief question, which relates to the clinical trial, because you talked about it. And we know that, you know, it typically takes such a long time to decide about the, you know, a treatment regime with traditional kind of FDA clinical trial pathway. And I know there was a lot of work trying, you know, as soon as possible to kind of determine the outcomes and to change the landscape so that the patients can benefit from the treatment sooner. Can you please comment on the changes that happened, how the data was used, and where would you want to see some algorithmic solutions which are beyond what people are doing?
So I think it's right to say early on in March and April in the United States, people were using unproven therapies like hydroxychloroquine, and relatively rapidly, people pivoted to doing randomized trials. So basically the months of April and May were largely devoted to randomized trials. Over 1,000 people were recruited into an NIH trial called the ACT trial, which is the trial that showed benefit of remdesivir. MGH was one of the top enrolling sites in the country for that. But it proved that you can do a clinical trial in a pandemic. In fact, not only can you do it, but you should do a clinical trial in a pandemic, because otherwise one makes no progress. We were making zero progress in March until these trials came online. And then we made relatively reasonable progress with the data for remdesivir, and then in parallel, the data that came out of the UK, where they enrolled in their National Health Service thousands of people very, very quickly. So again, the one lesson of COVID and of Ebola and of HIV is those who doubt that you should do a clinical trial in a pandemic are completely misguided. It's actually, that's when you need a clinical trial, is during a pandemic, because you don't know what works. But the data that has come out of that has informed some clinical decisions. We don't know now how to combine things. The National Institutes of Health, I'm on a panel, and we have struggled for the last 6 weeks trying to figure out, should someone on supplemental oxygen get remdesivir, dexamethasone, or both? And the trouble is we actually don't know. We really don't know. It's very much theoretic. It's very much expert opinion, and we've struggled with that. The other thing that I can say is combination therapies are now coming online. The same NIH group that did the remdesivir trial has just completed a trial, the data to be released in the next month or so, of a combination of an anti-inflammatory baricitinib, which is a JAK kinase inhibitor, with remdesivir. So an anti-inflammatory with remdesivir as compared to remdesivir alone. So now you have some data that you can do comparative of combination versus a single drug. I've worked in HIV for a long time. There was tremendous pressure in the 1990s to approve drugs for HIV, and the FDA and the NIH resisted that pressure. And that's what led, I think, to the combinations. You know, people said we need to do comparative trials. And now HIV is very much a controllable disease because of those trials. The dedication of the clinicians and the patients in the 1990s is why we're much better. And the same will happen with COVID, for sure. It just will happen a lot faster because there's so much more COVID, and it's a very acute disease, whereas HIV is much of a slower disease. So I don't know if I precisely answered your question. I hope I did.
This is great. I think you gave us a lot of interesting ideas and really food for thought. Yeah. Because the other conference that we're organizing actually talks about drug discovery and our efforts, and we are working a lot actually on combinations. Good. By the success in HIV. So it's really, really interesting, but I would stop monopolizing the time. Thank you, Raj, and I will move to Guillermo. So Guillermo, you have a unique perspective of, kind of comparing between the US healthcare system where you worked for many years and being physician in Mexico. And again, looking at the huge discrepancy in care between people who have access to the best of care and people who don't have access to great care. So based on this rich experience, can you tell me what are the technologies that you really wish you would have had with AI and you so far don't have them?
So I think my wish list will start with devising an algorithm to discriminate those that will get very sick and those who will not get very sick. And I think that's a fundamental question, because I think that would be a way to really concentrate effort, resources, vaccine strategies among those that are more susceptible. And I think that that's a major area of work, and it's clearly a big unknown. And some of us that take care of patients every day, and even though I'm not daily taking care of COVID patients, I consult on patients that are COVID because a large proportion of those have cardiovascular disease. Clearly, there are individuals that look the same to us. And I'll give you an example, as Raj was commenting on 3 different clinical scenarios. If I may have 2 individuals that are in their mid-30s that are somewhat overweight, not obese, with no overt diabetes, and one of those in the ICU, 3 weeks on a ventilator, considered ECMO, eventually died, and the other one basically had a cold and went home. And it's very difficult to dissect what are the factors that determine in an individual patient the risk. Now, so I think that that's a big issue for people in countries where the resources are unlimited, because we don't have the ability to follow patients at home the way you may be able to do it in Europe or in North America. And just for the people to have an idea, in a country like Mexico, where government provided services account for 90% of the population, they cannot make a phone call to a physician and the physician answers. I mean, if they need something, they have to go to the emergency room and wait for hours before they get seen. So if we had a way to put a patient through a machine and then define, this is going to be okay. So, or his risk is low, then he may be able to work, he may be interacting with others. And if he were to be sick, we will be less concerned about those. And maybe we'll selectively isolate those that were at higher risk. Now, in some way, we tried to do that by very broad criteria: age, diabetes, comorbidities, associated cardiovascular diseases, malignancies. But the reality is that even those are really difficult. We just had a patient last week that is 80 years old, was in the hospital for 2 weeks on a ventilator, and was discharged home totally fine without any cardiovascular or pulmonary sequelae. And then we have kids that are in the ICU for 3 weeks, survive, and have pulmonary fibrosis to the point that they are crippled, that may need lung transplantation. So the disease complexity is such that my wish, and it would be very appropriate for everyone, but even more critical for places where resources are so difficult to obtain, that if we had a better, simple way to predict risk. Now, is that going to include just biomarkers? Is that going to include just genetic predisposition based on family history? Or do we need to add sequencing of particular proteins of inflammation or other associated factors that may potentially give you the clue? So I think, in a way, a very large effort to do a more precise risk allocation of an individual would be ideal, which will help populations, will help better care, and it also provide more reestablishment of the economy, which today in the US, of course, it's an issue, but it's even more dramatic in places like Mexico, where if you don't work for 2 weeks, you don't really have anything to put on the table. And that's the reality of a lot of Mexicans, where be able to go back, it's so important. And what are the protocols for reestablishing back the economy? Really, we don't really have them. or we just don't take advantage of all the potential information that's available to better stratify that.
Thank you very much. I just want to ask a question because both you and Raj were talking about kind of not knowing what to do and which therapy to give. I'm just curious because clearly the machine learning model would learn if there is a signal in the data, correct? So assuming that you guys are collecting biomarkers, you Guillermo collect one set, or the doctors kind of have, I don't know, imaging or blood work or whatever. Raj, you also collect some data. What do you say is that over the 6 months of pandemics, you as the doctors develop your own intuition that kind of tells that you can better now discriminate between, these cases that are going to escalate, or you're still puzzled? I'm sure there are cases that it's always puzzling, that you gave examples of those. But I'm talking about the majority of cases, because if the signal is not there, it doesn't matter which machine learning you do, it's not going to be capable of learning.
I didn't recount this story, but I have a healthcare worker who's in his early 50s who initially tested negative for COVID, probably falsely negative, and then was tested positive. And once he was positive, he was home. He was not very sick, but when I spoke to him on the phone, he seemed short of breath, which concerned me. And he had an oximeter. His oximeter was giving good readings, high oxygen saturations. But literally, I then called him over the next 3 days to make sure that shortness of breath wasn't changing. And he did fine. But just like was said, if I had some better way other than relying on how he sounded on the phone and maybe his oximeter, I could have had a better weekend, basically, because I wouldn't have been as concerned about him. So we have developed this intuition, or rule of thumb, I guess you'd say, that shortness of breath, which usually on average occurs about 8 days after symptom onset, usually portends a poor outcome. And that can be seen with a low oxygen saturation, maybe the impaired gait with that Emerald project. But it's not very precise. And the data on outpatients is not as good as the data on inpatients. De facto, we don't collect as much data, at least to date, on outpatients, because we're trying to avoid them coming into the hospital. I agree. I agree with Guillermo. That's very much a need. This 90-year-old that I hearken to, should he get a monoclonal antibody? I really don't know that right now. Should he come to the hospital? I don't know that. And if we could collect 100 90-year-olds or 1,000 90-year-olds, maybe we could get some information on that. One thing I will say is, even though age is an extremely strong predictor of poor outcome, even most 90-year-olds will recover from COVID-19. A good proportion won't, because there's a very strong relationship between age and poor outcome. But we don't know which of those 90-year-olds will recover and which ones won't.
I'll just extend the comment, Regina, just by, we had an interesting problem in our hospital, because at the beginning, we started seeing the surge of cases in around June, which is probably later than when it occurred in the US. So at that time, we had access to anti-IL-6 therapy. Wasn't clear what to do with steroids, but ICU physicians are divided in their biases towards the use of steroids or not in very critically ill patients. So the data was coming up, and there was confusing data, because at the beginning, anti-IL-6 therapy, tocilizumab, was not good, and then good, and then bad again, and then good again. But the doctors that believe on it, and they had an intrinsic bias that it worked, they kept insisting that they wanted to use tocilizumab, even though the data wasn't really supporting it. And then at the end, now it looks like it's probably better. And then, so I am totally confused, but there are physicians that always had an intrinsic bias towards the use. So individual observations are useful, but your bias is so difficult to put together with reality sometimes, that I am always very concerned when doctors say, oh, in my experience, blah, 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 because at the end of the day, you're just putting all your bias into a particular strategy, and it's hard. Yeah. So lots of data with multiple sites, with a lot of intelligent people, will provide a better answer that individual bias. The bias, I think, can establish hypotheses that should be tested. But many of us on an individual case sometimes forget a lot of the data when we try to make individual decisions. But anyway, so I think that I just wanted to put that in perspective for those of the people in the audience that don't do clinical work or have not been involved in making clinical decisions at the level. It is challenging.
So thank you very much. And with that, I want to kind of have a question for the person who represents organization in the United States, which is like the major powerhouse of data and which gives recommendation to us what to do based on all their data. So Dr. Wong. So Karen, my question is, given your very special position, you are not just a member of CDC, you are actually the one who is leading the data science efforts. And I don't know if you listened to my opening when I talk about William Farr, who was able to do some important discovery regarding cholera just by doing the counts. Hopefully that's not a CDC is doing today. So my question is, how does CDC today utilizes the power of AI to get better insights from the data, if it does?
Yes, thanks. So I think, at the beginning of any new public health crisis, people are looking to CDC for answers and guidance. But the truth is, at the beginning of this pandemic, we didn't have much more or better data than anyone else. It just didn't exist. And so I think what we've tried hard to do is to use AI and other advanced analytic techniques to make the most of what we do have, while also being able to acknowledge the uncertainties in the data, because there's, I think, particularly if you work at a place like CDC, there's tremendous danger in maybe making suggestions that turn out to be wrong later, right? And so to give one example of how we were trying to inform guidance and practice in that early data sparse environment, I think early we were just inundated with emails and phone calls from clinicians and members of the public, asking us questions about things that we just didn't know and nobody knew. And so one of the things that we did in the absence of traditional epi and clinical data is that we sort of rapidly stood up these NLP tools, natural language processing tools, to understand these informal text data that we were receiving from the clinical community. And I think, even if it didn't sort of give us, maybe hard answers, this helped inform our guidance and research priorities by being able to understand what it was the clinical community needed to know. And I think it did help us pick up some early signals from clinical observations that might otherwise be lost or might not have come out until later, when you depend on, the astute clinician being the sentinel for a certain disease manifestation. And so we also, we tried to do this sort of rapid clinical data collection on the early cases, and some of the things that we've been working on are being able to identify these different clinical phenotypes, as my colleagues have mentioned here, to better understand the heterogeneity of disease, because there is tremendous heterogeneity and it's not very intuitive. And then, as some of the other presenters have said throughout the day, identifying predictors of severe outcomes. And so I wanted to raise, I think, 3 open questions for the community. I agree with Dr. Gandhi that there's this big question around, who goes down this hyperinflammatory syndrome pathway. And this is something that was first recognized in children, but may also occur in adults. Can be hard to distinguish from, particularly in adults, from just kind of acute COVID and kind of, somebody who has a lot of comorbidities at baseline versus somebody who is truly going down like a different physiological path with the inflammation. And so this has profound implications for treatment, but I think that it also kind of remains to be seen whether this may have implications for things like vaccine response and reinfection, which are going to have, huge public health impact. And even though it remains to be seen, it sure would be nice to get ahead of it, to know more about who develops this inflammatory syndrome. Another aspect of the COVID phenotype, I think, are the long haulers. So those patients who, maybe recover from their acute COVID, but have these chronic symptoms that last, for weeks or months. So who is it who develops these sequelae? And it can be young, healthy people. So it's not necessarily intuitive there either, and that has implications, I think, for the burden on the healthcare system and for what we will have to look forward to as a public health community in terms of the disease landscape. And then finally, I think a question that is out there and that is going to become important is the question around sort of reinfection and waning immunity. So it's certainly clinical implications, but also public health implications. This likely happens in a heterogeneous way and maybe not in a totally intuitive way again. And so, so if we knew sort of what the course of waning immunity might be and what the risks for reinfection might be, that's going to require some marriage of clinical and clinical data and biomarkers and probably some epidemiologic and exposure data as well. But I think that this could help us target prevention strategies on an individual patient level, but also on a population level. So I'll pause there and see if anyone else wants to comment.
Oh, thank you. And before we are moving to Gabi, I have a very brief question. So how much data that can help us to answer this, fundamental questions that you brought up? CDC is ready to release to the AI research community to help with finding answers to the questions.
Yes, I mean, I think this brings up an important issue, is that there are different kinds of data for different questions and different quality of data for different questions. So I think as Dr. Stoltz mentioned earlier, there's tremendous value in the small data that's very carefully adjudicated, particularly for answering some of these very complex clinical questions. And then I think the places from, say, electronic health records, where you might get larger volumes of data, just don't have the level of resolution that you might need to answer something that's very nuanced. And the other thing is that I think for all of these data, some of the epidemiologic aspects are not necessarily in medical records. So information about a patient's exposures, behaviors, symptoms, these aren't always well documented in health records. And I think that's a place where natural language processing may be able to help. But I think it is very tricky. I mean, even for us at CDC, people may think that we are sitting on a mountain of data, but that we often have trouble getting access to the right data and being able to put the right types of data together to answer the questions that are out there.
Thank you. Thank you. And now I want to ask a question, Gabi, who is quite different again from the medical doctors that are present, in terms of she is, both a patient and somebody who decided to change, patient outcomes and create, really help to the population, which typically overlooked in medical system, which is underprivileged women. And my question is how the reality changed for the Protea Institute and for the treatment of underprivileged population with breast cancer, which is representative of other chronic conditions during the COVID time, and what steps are you taking?
Sure, thanks, Regina. So I think, unfortunately, the reality is that I think we will have an epidemic of cancer, after the COVID. And I think for you to understand, I need to explain how the Brazilian health system works. Basically, of the 210 million Brazilians, the population, 75% or 160 million people do not have private health insurance. They must rely on the public health system. The problem is that the public health system does not have the capacity to treat all the Brazilians on a timely manner. So the public health system actually doesn't have enough public hospitals to treat all these people. So it does partnerships with private and philanthropic hospitals and asks these hospitals to treat patients from the public health system the problem is that it doesn't pay even the cost of those treatments. So those private and philanthropic hospitals, they limit the number of patients that they treat from the public health system. So that's already a problem pre-COVID. What happened was that in breast cancer specifically, the statistics are terrible. They're very dire. As you probably know, 1 in 8 women, that's a global statistics, will develop breast cancer over their lifetime, 1 in 8. 12.5% is the most common form of cancer for women. And the mortality rate in Brazil, unfortunately, today is 25%. So 1 in 4 women are dying of breast cancer within 5 years of the diagnosis. This is very high. It's much higher if you look at that mortality rate in the rest of the world is between 5 and 10%. So 90 to 95% of women that are diagnosed in breast cancer, and that's because most of them are also diagnosed early enough, they have 90 to 95% chance of cure. So again, that is an enormous problem in Brazil. The statistics is 42 women are dying of breast cancer every day in Brazil. This is a reality. So there are 2 reasons. One, the diagnostics in the public health system is late, is much later than in the private health system. For example, one reason is the government only pays for the mammography starting at 50 years old and every 2 years, while women in the public health systems, like for example in the US, they tend to start doing the mammography at 40 years old. Also, there is a lot more women that don't do the mammography in the public health system than in the private health system. So the 2 reasons are, one, the diagnostic is late, as I just explained. Just to give also another statistics, in the private health system, about 17% of the breast cancers are diagnosed above stage 3 and 4. In the public health system, that 17% is 37%. So again, an enormous difference. So the second reason why the mortality rate is so high for breast cancer in Brazil is because when women are diagnosed, they go and they wait to start treatment months. More than 66% of women wait at least 2 months, but there are women that wait 6 months, sometimes a year to start treatment. So in other words, you have your diagnosis and you're gonna wait. And again, that's why I started the Institute, Protea, to really start giving access and accelerating the treatment for these women that are diagnosed. So what COVID has caused, I'll give you a couple of statistics. So COVID is gonna make all this worse, unfortunately. Why? Because, for example, it's estimated that since March, more than 50,000 cancer diagnoses were not given, because for the same reasons that Connie explained in her presentation. I mean, women are not going, they were afraid of going to the public health system. The way it works is that the women from the public health system, they go to the public health clinics to have their first consultation. And then when there is a suspicion, they are sent to a hospital, either a public hospital or a private or philanthropic hospital that has partnered with the public system. So again, in breast cancer, screening, consultations, and mammograms have been suspended, literally suspended since March of this year in the whole public health system. Recently, they have been restated, but they have not been reestablished as they were in March. And so it's also estimated that 43% of the patients that were undergoing treatment, 43% of patients that were undergoing treatment had either suspension in the treatment, in other words, whether chemotherapy or whether surgeries or even consultations. So that's why, I'm telling you that unfortunately COVID is going to cause, once COVID is over, an increase. It's estimated, specifically in breast cancer, that it will be an increase of 30 to 60% in new cases. Also, if I can give you, Regina, a specific number for the hospital Santa Marcelina, where Protea Institute helps and treats and increases the number of women treated from breast cancer, we have been growing a lot. And in fact, we had plans to grow significantly this year. So just to give you some numbers, last year we impacted 1,417 women in that hospital that we partnered with. And that's a significant increase from the year before. And this year we were planning to increase, originally before the pandemic, 36%. Now what we're seeing, and just to give you some numbers, we had in January and February, we had 127 on average women per month that we were impacted with treatment, with exams, with consultations, with everything as it relates to breast cancer treatment. That 127 average number fell to 76 in March. We've already recovered, but we haven't reached the 127 yet. So just to give you a number, again, if in the first 2 months of the year, we will impact 127 women per month, on average since March, we have impacted 89 women on average. So from 76 we grew, and the average is 76. So a 30% decrease in the women that we were impacting on a month-by-month basis. So, what are we doing to change this? I would say in the short term, as it relates to COVID, we are trying to increase, the word and the awareness for the women that live in the area that the hospital operates, and tell them that they should go for the screening, they shouldn't be afraid, and to try. So we are hoping that 30% decline that we've had, it's going to increase, and we're going to end the year just 11.5% down from last year. So again, if we look at numbers from the 1,417 women that we impacted last year, this year, assuming that we have started to recover, and we have, in fact, in September, we're going to end the year 11.5% down. And again, this is much lower than we were hoping to do pre-pandemic, where we're going to grow 36% on a yearly basis. The second thing that we're doing is obviously long-term. So what we have seen with this problem of the very high mortality rate is the late diagnosis. And that's where your technology and Connie's technology comes to make a huge difference. For example, in the hospital that we're treating, we are seeing that half of the women that arrive, they arrive with advanced cancer, breast cancer. So that's why, being able to diagnose early, it's going to be paramount for us to really be successful in reducing the mortality rate. So for that, we are, thanks to yours and Connie's willingness to really make a difference with your technology to the women in need, to the women that really don't have the access. We are in the process, as you know, to validate your technology in Brazil with two institutions. One is called Barretos Hospital, is a philanthropic hospital in the interior of the state of São Paulo that basically treats 100% patients from the public sector. They have an enormous amount of almost 250,000 mammograms a year that they do. And so we're validating the technology. We're hoping to have, a very powerful validation with the Brazilian population very soon. And also, we are also validating with a diagnostic imaging NGO called FIDE that basically does all the diagnostic imaging diagnostics for several public hospitals in the city of São Paulo, in the state of São Paulo. And we are also hoping to validate your technology with the Brazilian population, those two institutions. And once we do that, the big dream is that we're going to go to the government with your technology and say, look, this is the difference. This is how earlier we are diagnosing breast cancer in Brazil. And therefore, we need to implement this in the overall public health system. We can hear you.
So, sorry, I just want to say thank you very much. And actually visited both of this hospital a year ago, pre-COVID time, and definitely they can benefit from the technology. And if we are operating in the case where our resources are limited, where what Guillermo was saying, what Gabi was saying, that in places where really the capacity is greatly limited, even at MGH, having AI to predict who really needs it and who should go first in line on getting treatment, I think that's where AI across many diseases, breast cancer is one of them, but many others, would really help. So now I want to go to a few questions that our audience asked. So there is the first question, I would say it's kind of AI question, I would answer it very fast. In regards to PI, are you using blockchain along with AI to create and track identities? To the best of my knowledge, and if other PIs are here, they are welcome to jump in. I don't know of any use of blockchain, in our MIT work. So let me go to the second question. You had mentioned that there are several treatment options that might be used in combination. How well understood are their interaction, and how distinguishable are their effects to see which option is having which effect on a high level? So I just want to you guys jump up and decide which one answers, but let's just keep the answers short so we can go through all the questions. So go ahead.
In terms of, this is Raj, in terms of interactions, we do know some of the drugs don't have pharmacodynamic or pharmacokinetic interactions. That is, they don't cancel each other out in terms of drug levels. In terms of how they work, we do have a general idea of, which ones are antivirals, which ones are anti-inflammatories. Although a drug like baricitinib, which is the one that's in this 1,000-person trial that the NIH is doing, is an anti-inflammatory, but it might also have antiviral effects. So I think the real question is, when you don't have data, can you mix and match these in a way? If you have a profile, an inflammatory profile, is that the time to add an anti-inflammatory, or is there some other way to, more rationally combine these agents that have different mechanisms of action? So I'll stop there.
Let me go to the next clinical question I would ask. I would read it out. Is the O2 need curve due to lung damage? There is a correlation of need to intubate with factors like the patient weight or had allergies? I mean, this is definitely beyond, it's too clinical for me. Guillermo or Raj, do you want to take this question, or Karen? So I guess the question is if the need to intubate correlates with vaping and allergies of the patient.
Oh, okay. I mean, I think in general, a pulmonary form of injury may put you at a higher risk, but I don't know that there is a particular association. With vaping, there is a lot of injury to the lung, and there's been more talk. But I think if you look at the factors that are associated with the risk of death, all of those are associated with a high risk of innovation. Because in most places where you have access to care, almost every individual that's cared for in a hospital before dying goes through intubation, unless for specific reasons of avoiding, the families or patients or the doctor recommend against it. But in general, the risk of factors associated with death are also those that are associated with the need for intubation.
Thank you. So there is a question which is on the AI side, but please feel free to jump in. Apart from models that use clinical data, has there been any AI work on developing causal reasoning models based on knowledge of biology, knowledge of drugs, and whatever is known about the coronavirus? So we would actually, in our conference, is going to be in a month, we will talk about like deep learning models. Some of them are causal models that look in this effect. I would say that currently there is like two classes of models, which unfortunately are not combined, as far as I know at least. There are some works which use AI or use most traditional statistical methods that really operate in the phenotypical space, which try to predict outcomes and other things. And there is also work which relates to really understand, the structure of the drug, the targets, and how they operate together. I haven't seen at least something that pulls the two together. First of all, it's extremely complex, but also I don't think there is we have now the right data to combine the two. So Guillermo, I've seen a question before for you that explicitly asked which data is your hospital collecting?
So we created a repository of clinical data. We have all the biomarkers, of course, the conventional laboratory data, and we're collecting blood samples to do sequencing. We really have not done the sequencing, but we have protocols approved by the IRB, as well as collected. I'm gonna lie to you, if, I don't know, we have over 1,000 samples collected for DNA sequencing, which we eventually want to embark on.
Great. So then there is a question, I guess, to all the members who are MDs, who are real doctors. When are you prescribing medicine to patients, do you take into account their possible polypharmacy side effects? I guess you do, no?
You do. I mean, it's a limited number of drugs thus far. Maybe I'll give the example that was mentioned of tocilizumab. That's an anti-IL-6 blocker. It doesn't have direct interactions with dexamethasone, but they both are potentially immunosuppressive, and they both could lead to increased infection, although the randomized trials to date have been somewhat reassuring on that front. Yes, we do. We take into account whether drugs are interacting with each other, as well as whether they have overlapping side effects.
I may add, just Regina, for the FDA, for regulatory approval of new medicines, that also it is a big issue. That we're working with a particular compound with part of our own development, and even though this compound that we're using that has anti-inflammatory effect, the interaction with a lot of the other medicines that a patient that's diabetic with cardiovascular disease or pulmonary disease has are totally unknown. There's a big area of concern for the FDA, new molecules, new drugs in the setting of patients that take multiple medications, like someone with heart failure or pulmonary disease, COPD, or diabetes.
So thank you very much. We just arrived to the end of the panel, and I would like to thank on behalf of all the listeners, and definitely on behalf of myself and JClinic and MIT, really thank you. I'm really thankful that you took time out. You were super busy schedule and caring for patients, doing great things for patients like Gabi does, and talk to us. I hope that it illustrated the type of problems that we on the AI side needs to be addressing. And I promise that I will follow up with different participants and see more what kind of data we can use, and we will publicize it on the JClinic social media and website. So not only MIT, but hopefully others can benefit from the data and eventually help patients. So with that, thank you very much. And thank you for all the participants and listeners who stayed with us. So thank you.
Thank you, Regina.
Thank you for organizing.
Pleasure.
Thank you.
Thank you.